Hello, I'm Jim Brewster, Education Coordinator for FamilyTreeDNA, and today we're going to be talking about a Big Y tool called the SNP Checker. And this is a really powerful tool that can give you a lot of in-depth analysis into your Y chromosome. It is a bit of an advanced topic and an advanced tool, and today the concepts that we're going to be talking about are going to be based on SNPs, mutations, haplogroups, and the YDNA reference sequence. So if any of these terms are unfamiliar to you, I would recommend pausing this video and taking a look to learn more about those. There's lots of great resources online. One that I would recommend, a bit partial to it, is the FamilyTreeDNA Help Center, which can be found at help.familytreedna.com. And in there, you'll find articles that address each of these concepts written by yours truly. So, first off, this is a Big Y tool. So to get to it, you'll need to have Big Y results. And this tool can be found under the results section here on Big Y on your main dashboard. And when you analyze the results, the end report is going to look something like this. And this can be kind of overwhelming. So first I want to reassure you, don't panic. We're going to cover all of this, and it's really going to help boost your genealogy. Now, one way that you can think about this is as if it's a map to your DNA results. And just like any map, there's a lot of information on it, and it can take a little bit of getting used to understand all that information. But once you do, it can really be a powerful tool to help you get to where you want to go. So, in order to fully understand the map, you have to understand a little bit about the geography. And in this case, the geography that we're talking about is the Y chromosome. So to put that into perspective, here is a picture of your chromosomes. Now this is an image called a karyotype, and it lists the human chromosomes. And you can see here that they appear in pairs. And that's because for each chromosome, you have one copy that was received by your father and one that was received by your mother. So it's a chromosome pair. So this can, it's a little hard to read because this is an actual image. So here, let's take a look at a diagram of it. Hopefully this will be a little bit easier. You can see that each of the chromosomes are numbered and they each appear in pairs. Now, today we're obviously focusing on the Y chromosome, and that's a special type of chromosome called a sex chromosome. It's what determines a person's genetic gender, male or female. So a male will have an X chromosome and a Y chromosome, while a female will have two X chromosomes. So because of that, the Y chromosome is passed down specifically through the male line, and it can tell us information about that specific male line. So let's zoom up on it here a little bit. And you can see that in the Y chromosome, at least in this diagram, there are several different colored bands in here. So first off, what does that mean? Well, again, here's a diagram for it. So this first area here, it's called the pseudoautosomal region 1. Why is it called that? It's because it almost acts a little bit like autosomal DNA in that it recombines with the other chromosome. So some of the things here in the pseudoautosomal region on this end and the pseudoautosomal region on this end are going to slightly recombine with the X chromosome, and because we want to focus specifically on the male line, we're going to ignore those. This large black section here is referred to as inaccessible. It's sometimes called a noisy region. It's essentially hard to get an accurate read of it, in part because it can be highly repetitive, but there's other reasons. So the focus that we have here is this white region. And this consists of 23.6 million base pairs of chromosomes. Now, I did a rough calculation just on my hands right quick, and it is way more than I can count on my hands, is the size of it. So even though it might be comparatively smaller than some of the other chromosomes, it's still a very large region. And this is the region that we are targeting in our Big Y test. So when we target this area, we're going to map it out. And so it's really important that we understand what that map is telling us. So if we blow up just a section of DNA here, you've probably seen something similar to this, the double helix shape of DNA. And you can see that the two rungs, or the two strands that are in it, are connected by these little, almost like rungs on a ladder. And these rungs are made up of 4 chemicals. We call them nucleotides. We typically abbreviate them as G, C, A, and T. So you can see that on each one here, we have a pair, and we have it in a very particular pattern. Because of the chemical shape of it, G always pairs with C, and A always pairs with T. So these rungs are going to be an AT pair, a CG pair, a TA pair, and so on. So if we were to read that entire segment, essentially stretch it out and then read it from start to finish, we're going to get a long set of paired letters. And that's because we have on each location here, on each rung, we have 2 nucleotides that are paired together. So here we have an A on the forward strand, and here on what's called the reverse strand, we have a T and a T, because A and T always connect to each other. Here it's the same thing, a T and an A connect, or a C and a G connect. Now, because we know that they connect in that way, we can ultimately only focus on one. That helps cut down a little bit on the confusion. And then we can map it out. So put it on coordinates on it, just like we do a location on a map. So we can say that at location 1, we have an A, and we refer to that as the value of that location. So location 1 has a value of A. Now we only report the forward strand value because, again, we know that if there's an A here, there has to be a T down here. So just by reporting one, we know the other. And when we combine that together, we get something like this. So this is just a list of the locations on the chromosome and what the value is at the top. Now this is a standard set of values. This is a reference set. This is essentially what we refer to track any mutations or any changes that we find. Those changes are going to be specific to our paternal line, so we're going to want to focus on being able to have a standard reference to compare those to. So this is what's called the reference sequence. So on each one of these positions on the reference sequence, there is a specific value. And when you have a SNP, it means that you have a change on that value. So back to where we were here with the target region of 23 million base pairs, we're going to get a long string of this. And so we need a tool to be able to focus on the SNPs that we want to see and focus on the regions that we want to look at. So again, you access the Big Y results here through the main dashboard. There's other ways to get to it as well. And first, you're going to see something that looks like this. Don't worry about too much of this for right now. What I want you to focus on here is the SNP checker. Now, in this box, you can enter either the name of a SNP or the position. Now, mutations that are different from the reference value that are found, once they're found, they're given a SNP name. That's usually something like M269 or BY1420. It's some combination of letters and numbers. And that name designates what the position is and what the change is, or what the value is. So in the SNP checker, you can either list a SNP name if it's been discovered before in other test takers and given a name, or you can enter a position. And that is useful if either it is unnamed, if it's new to you, or if all you have to go off of is the position for something and you want to see just what that value is, whether it's a SNP or not. So when you enter that, for example, let's go ahead and enter here M253. Now this is the defining SNP for haplogroup I. And I chose this one because yours truly is in haplogroup I, which has been scientifically proven as the best haplogroup. So here you can see this banner along the top of the reference values and the locations, and it highlights the SNP that we've looked for. So you can see here that the reference value is C. So if I have anything different than C, it means I'm positive for that mutation or I'm positive for that SNP. So each one of these is a read, and we'll go into what a read is here in a moment. Each one of these is essentially a check of that section, and you can see that in each one, it picked up a T. In this next example here, you can see that it also read this section a whole lot, and it only picked up a T. So that means that I don't have that mutation. It means I have the same value as the ancestral value. So that isn't one that defines my haplogroup, therefore I don't have it. Now, as I said, each one of these is a read. So it's essentially like proofreading the book that is your Y chromosome. And just like when you try to proofread something, occasionally you'll miss a typo or you'll catch a typo. And so it's important to proofread something multiple times to make sure that you get the accurate read for it or the accurate result. So each one of these is a read through of that location. And that's really important because the more times that you read a certain location, the more confident you can be that you got the correct value. You can see here in this one lone read picked up a G here on this location. Checking it multiple times can be important to weed out false positives. If all we did was this one read and we found this, then we might think I'm positive. But it's really just a typo because all of these other ones didn't pick it up. So it can help weed that out. It can also help verify that you have a SNP. And the number of times that it's read is referred to as read depth. So we're going to talk about read depth and quality. Read depth is the number of times a location has been tested. The deeper the read, the more accurate we are in it. Quality we'll talk about in a moment here, but one thing I'd like to point out, just because again this can be a lot of information, is that you'll see that they're in two colors, and that's red or blue, and that's just a forward read or a reverse read. If you'll remember, there's a forward strand of DNA and a reverse strand, and if we know the value on one, we automatically know the value on the other. So that means we can read the reverse strand just as easily as the forward strand and know what the forward strand is going to say, because we use that forward strand as the standard. So the different colors just means whether it's going front or back. So here's an example of a read that doesn't have the best read depth. You can see that in this one, it was only covered a few times. But every time that it was covered, it was positive for that SNP. It got a nice clear read. So that's important. And that can verify that I'm positive for this SNP. So you might be asking, okay, well, why are there fewer reads here? Or why isn't every read just as long? Well, and that's due to the nature of testing. And there's a blog article by Dave Vance that goes into it, if you're interested. But basically, in a single read, it's going to miss sections. So there's going to be some sections that it will skip over entirely, or some that it kind of has spotty coverage for. And that spotty coverage is what we refer to as the read quality. So basically, the quality means that we know we're looking at the right place in the chromosome, that we know that we're on the right page. Because sometimes, the sections of chromosomes, they can look somewhat similar to each other. So it's sort of like if you're anything like me and you go on a hike in the woods and you don't bring your map because obviously maps and directions are beneath you and you don't need them, then you have a hard time sometimes seeing where you are. And you might think, hey, didn't we pass this tree already? Is this the same tree or is this different? Well, this is telling you that we're not quite as confident. So we did get some good reads, but you can see here that a lot of them weren't confident, so this is something that's taken into account when calling this. But whether or not you're called for a SNP, you can always look it up by the location. Which brings you to the next part of finding your SNPs, because I'm sure it's your own that you'll find most interesting. So on your SNP page, or on your Big Y results page, you'll see here that you have named variants. These are all of the SNPs that you have tested positive for, which have been named and placed on the tree. You can find the name, the position that it's at, whether it has been added to the global haplotree of mankind, as well as what the ancestral or reference value is and what your value is. I am positive for all of these SNPs because I have a change or in the value of the ancestral value. So I'm positive for that SNP. So I have a derived value or derivation of that value that's different. So this can give you all of that information. There's also a section here called private variants. These are ones that have not been placed on the tree, either because not enough test takers have tested for it or other reasons. But you can see here, just to blow it up a little bit, it only lists it by position because this is unnamed. It hasn't been found in anyone else. Once it is found in someone else, it'll be named and placed on the tree. But for right now, it's unique to me. And again, it lists the ancestral and the derived values. Lastly, we have matching. And these are the people who match you on these same SNPs. So it's going to here list the match name, whether or not they have a tree, the non-matching variants, so that means the variants that either you're positive for and they're negative for, or vice versa. And you can click on their name to find out more information about them, email them, and help boost your genealogy together. So this can be a really great tool to not only look up data, but to verify data and see how you compare to others on it. You can learn more at FamilyTreeDNA.com, and I hope you have a great day.